Okay, so the Rubisco and why is it a faulty enzyme? So I already mentioned what does it stand for. Um, Now, Rubisco is regulated and activated first and foremost by light. Now, however, it is a very kind of high maintenance protein because because of so many reasons. So let's start talking about those reasons. So I mean, this is a kind of complicated story, but don't blame me. First of all, it needs to have for the Rubisco to start to work, we need light. Then it needs a certain amount of hydrogens to be stored inside the lumen. Then it can only work if the stroma pH is around 8. It is activated in the presence of the carbon dioxide. It is also activated, so magnesium, the two-valent magnesium is one of the coenzymes which are going to activate Rubisco. And to make things even more complicated, there are other enzymes which need to be activated before Rubisco can go from its inactive stage into its active stage. So I have to explain to you this complicated story. Um, so here the target enzyme that's Rubisco. So let's start from the beginning. So we need to have all these conditions with the pH, with the magnesium, and I cannot remember what I said, but you see, we have light here, and then at the end of the electron transport chain, remember there is the ferredoxin, which will convert NAD plus into NADPH. So when that happens, then another enzyme will activate an enzyme which is called thioredoxin, and the oxidized state of thioredoxin will then convert Rubisco from its inactive state to its active state. So only then, in right here, is Rubisco ready to fix carbon dioxide. That is why, we see three major problems with Rubisco. So with all these things that have to be met, plus you have two additional enzymes which have to be activated before Rubisco, you can see that Rubisco has a very slow catalytic cycle. And then it actually has a very low affinity to CO2. So it gets kind of confused when there is CO2 and O2 presence in the air, because Rubisco doesn't differentiate this double bond between CO2 and between oxygen molecules. So to Rubisco, these and these, they're completely the same. So what can happen is that instead of carbon dioxide, Rubisco can accidentally fix oxygen. So when that happens, and I mean, of course, that is bad, but you see how instead of this normal case scenario, when we're going to have when we're going to have two 3PG molecules, instead of that, when Rubisco adds oxygen to ribulose 1,5 biphosphate, there is only one molecule of 3PG that's being created, and then you have this guy called 2-phosphoglycolate. Okay, so glycolate, it's actually toxic to plants. It is very toxic, and it can cause damage. Now, what we see here, right? So here we go again, another complicated biochemistry cycle. So what you see here, I'm actually trying to locate. Oh, there it is, glycolate, right here. So what are we seeing here in a nutshell? We are seeing three organelles. So we see chloroplast, we see peroxisome, and we see mitochondria, and then we see glycolate coming out of the Calvin cycle as an accidental toxic byproduct when Rubisco fix oxygen instead of carbon dioxide. And this is actually called photorespiratory pathway. Now, please remember this is bad, okay? This is bad for the plants because, as I said, glycolate is toxic. Now, the $1,000,000 question here is, okay, well, if glycolate is toxic, then why doesn't the plant just throw it away as a toxic byproduct? We don't see that. So, as a logical thing, we will expect, okay, glycolate, toxic, plants, throw them out. But this is not what we are seeing in the biochemistry. What we see is this. So there is like a lot of things that are going on in this three organelles. And what is actually happening in the photorespiratory way is we see all these three organelles, so the chloroplast, the peroxisome, and the mitochondria. Then we see all these intermediate amino acids, and most of the enzymes which are responsible to converting whichever amino acids and other types of molecules you see here, that's not their job. So the enzymes that are doing something else, they're actually hijacked to deal with glycolate now. So the enzymes involved are not specific for the photorespiratory pathway. They're just, they're kind of taken away from this job and they're told, oh, now you have to deal with this and go into the photorespiratory way. And then on top of that, what you see here is there is a lot of energy being used. So the $1,000,000 question is why? Why doesn't the plant just get rid of the glycolate, and then continue doing whatever plants do? Why? Why will the plants use energy, hijack the enzymes, and go through all these complicated biochemistry? What you are actually seeing here, okay? Why? And let's just go back to looking at the glycolate. So is there anything special about this molecule? You see anything? I don't really see anything special. It has a phosphate group, and like this is where the oxygen is being attached to. So like really, but when you go into details, what you see here is plants are desperately trying to save those two carbons from throwing them away. Okay, so photorespiration is really a plant desperate try to kind of try to save those two carbons from just be wasted away. Okay, so that's the answer to the $1,000,000 question. And actually, Rubisco is so bad at fixing carbon that the ratio of carboxylation when you fix carbon to oxygenation when Rubisco is fixing oxygen is 3:1. So yes, it is fixing carbon, but like from time to time, it is going to fix oxygen. So with all this story, you can see how Rubisco it's kind of, there is like something seriously wrong with this enzyme, and we will later come to the conclusion what is actually happening. Now, plants that cannot photorespire, so plants that they cannot go through all this complicated cycle to save those two carbons, they are much more susceptible to damage when either there is a lot of light or when there is little light. So right here you can see this is a cannabis, which is a typical example when you sunburn or when you light burn the plants. So too much light is also bad for plants. And of course, when you grow plants in shade, they will also do bad. So under low light conditions, like Rubisco will start to kind of twitch and and start to accumulate and fix oxygen instead of carbon dioxide. The only good thing that comes out of this PCO cycle is that it is going to kind of accumulate some of the amino acids, especially glycine and serine, into the metabolism, and especially, it is going to minimize the damage caused by the photooxidation. So when we are about to calculate the overall apparent photosynthesis. So the overall photosynthesis, we need to kind of subtract the respiration from the mitochondria and also subtract the photorespiration from the gross photosynthesis. Now, once the plant has created those glucose, not all glucose will go into sugar and starch. There is something that is called pentose phosphate pathway, or botanists, they like to call it oxidative pentose phosphate cycle, or OPPC, I just like to call it in a shorter way, pentose phosphate pathway. So you will see that some of the glucose that's being created, it is not going to go into, creating some of the basic sugars and starch. So some of that glucose will be converted into two-step process into ribulose 5-phosphate. So please tell me, where do you see the usage of ribulose 5-phosphate? Where? For what we are going to use this sugar? Now, a pentose phosphate pathway also happens in animal cells. So when glucose, of course, we cannot create our glucose. If we want glucose, I usually eat chocolate. So, there it is, the glucose from whatever chocolate I ate is going to go inside, and then it's going to, go into glycolysis, and then I'm going to respire. But some of that glucose, it is going to be converted into those ribose sugars to make DNA. DNA, deoxyribonucleic acid. So remember, the sugar in the DNA, it's a ribose. So some of the chocolate that I eat will go into growth. That's why kids like sugar so much. I hope you don't hear my daughter in the background, but that's one of the reasons kids like to eat a lot of glucose, because they grow, and some of that glucose will be converted into DNA. So the same thing happens in both plants and animal cells, the pentose phosphate pathway. Okay, now we have to move on, talking about two types of plants we have here. So all these cycles that I have explained, these are C3 plants, but we have two more types of plants, which are C4 and CAM plants. So C3 plants, here you see, as soon as the amount of oxygen increases, okay, rubisco is starting to fix oxygen instead of carbon dioxide, and then you will see a sharp line, so a drop in the photorespiration, in the photosynthesis. So more oxygen, meaning the rate of photosynthesis will rapidly fall within the C3 plants. Now you have C4 plants, and they look really kind of special because their photosynthesis rate will kind of remain the same regardless of the amount of oxygen that is in the atmosphere. So let's try to answer why is that like that.